Yeah.
Like a pundit square, right?
Yeah, that's right. So the genotypes, genotypes are the ones. Genotype is an expression that shows the alleles of the person. So this is genotype and phenotype are the two terms that are used to describe some of those traits. Genotype means alleles of a person. Alleles of a person for a given trait. For example, the genotype in this case will be. You can actually take any letters to describe it. If you are trying to describe the eye color, you can say this is this eye color. That shows that this is brown eye color. But if it is a dominant, a recessive, then you have to change one to the lower case. So this is dominant, this is recessive. The big B is dominant, so it controls the eye color. So this is dominant, this is recessive. When you write them like that, as a BB, so to describe an eye color, then this is a genotype. So genotype is an expression to indicate what is present at the genomic level, at the DNA level, at the allele level. Phenotype is what you can see, in a personal, in a kind of appearance, physical appearance. So eye color itself is a phenotype. You can see, or height of a person is a phenotype. The shape of the lobe is a phenotype. So it is a physical expression, what you can see. The genotype is based on the genetic condition, which is contained inside on the chromosomes inside the person's cells. So genotype cannot be seen. It can be estimated. It can be determined with a test. Phenotype is something that you can see, and you can see this person is 5 feet 2 inches tall. So that is its phenotype. Or if you see a flower, you can say this is red flower, this is blue flower. That is its phenotype. Eye color is the phenotype as well. Some traits are linked to the sex chromosomes. Those traits which are present on the sex chromosomes are called X-linked chromosome or sex-linked traits. And since females have 2 X chromosomes, usually those traits are passed from the mothers to their sons and to their daughters. There are several diseases like that. Hemophilia is one disease like that, which is the failure of clotting, failure of the clotting factors. So the blood does not clot. If it starts to run, it has to be kind of treated differently. Naturally, it does not stop. Another condition is kind of color blindness. Color blindness also comes from X chromosome. Then there are many other ones too that are actually passed. Or baldness, pattern baldness also comes from X chromosome. So all the bad things that are given to the sons come from the mother's X chromosome. So that's why it's important to have actually X chromosome, which is very healthy. So X chromosomes, the sex-linked traits can also be followed through the same type of Punnett squares. So in those cases where those Punnett squares are run, so in this case this is a male, so male that produces X and Y chromosomes, the sperms with the X and Y chromosome, female that is carrying some type of disease. For example, this is hemophilia. So these are this is the genotype, has 2 X chromosome, and 1 X chromosome has big H, which is the dominant. So this is the one that causes the hemophilia because it's dominant. The other chromosome, the other X chromosome is recessive for this trait. So its genotype is X and X, and one is, or you can also write it down with H and H.
Yeah, put the
So since we know that this is sex-linked trait, so you can also write down big H and little h. So big H would mean that this is on X chromosome, a dominant allele, and this one on the other X chromosome, which is the recessive allele. So this is its genotype. The phenotype is it has hemophilia, or it's the carrier of hemophilia. The male male is normal because it has a dominant allele, so it does not have any recessive allele. This one has a recessive allele, so this means that the mother is a normal carrier of this disease because she has this little h on X chromosome. This one does not have, this one has only one X chromosome, which is a dominant allele, shown with the big H here. So when the sperms are produced, one sperm does not have X chromosome, has only Y chromosome, so that's the safest one. The other one is a dominant allele. So in case of female, she produces two gametes, also one with the big H and the other one with the little h. So when the probability, when the first child is born, the probability is the first child has both dominant allele because came from the two dominant alleles coming from the sperm and from the egg cell also. The second one, the probability is the second child could be another female. So this is also a carrier because at least there is one dominant allele there, so she will not be the one that shows the disease. So she's not showing the phenotype, but she's just a carrier also. Another child, that could be even the first child, the probability is that will be a female, a male, because it has a Y chromosome, and because of this dominant allele here would be a normal male, dominant normal male. And the fourth one will have this recessive allele coming from the mother's side. So this boy will get this recessive allele from the mother's side and Y chromosome coming from the father's side. So these are the four probabilities. It could be the first child that they have is this one, or this one, or this one, or this one, or it could be any of the child, could be the first one, or it could be any of the child as the last one also, because these are all based on probabilities. It could be the first one, or it could be the last one, or it could be never. So it doesn't mean that there is a guarantee that that the first child will have this disease or the last child will have this disease or not. So when you analyze this data, which is the Punnett square itself, you analyze that this is a dominant situation where this female is dominant, this male is dominant, so normal. This female is a carrier because it has one dominant and one recessive allele, and this male is the only one which has this recessive allele. So this one will be the one that will be that will show this disease.
Oh, so it's not like with the what you call it? That's the father. Does what's the disease that both parents have to have it?
Oh, this is dominant, like in case of Alzheimer, for example, or any other diseases like that, which are pretty nasty diseases. So in that case, since those are dominant diseases, this is a recessive condition. So that's why only 1 out of 4 will have it.
Okay.
And usually hemophilic are usually seen actually in case of males, because if somebody is hemophilic, for example this one, so probably will not survive for too long. So like this is the condition which was seen in those Russian czars. So when they were captured back in 17, when all of them were captured, and they had only one son surviving at the time, and actually had hemophilia. The 2 or 3 daughters that they captured, including that princess, what was her name? Anastasia? Anastasia was one of those daughters, and all of them were actually executed at that time. But one of the son who was alive at that time had hemophilia and was so very weak and could not really do anything. Even if he was not weak, he was not going to be able to do anything. So this is how these type of recessive diseases are carried from one generation to the next generation. For example, if this disease was a dominant disease, then this person will have it, this daughter will have it, this daughter will have it, this son will have it, and this is the only son who will not have it. So for example, if this was a dominant disease, then it will be completely reversed. So if it is albinism or things like that, or other diseases, then only one that who will not have this disease will be this one child, one son that they have. Everybody else will have this disease. Then, so this is the condition, what we have seen so far. Those are the conditions based on the dominant and recessive alleles. Then there are conditions which are not dominant or recessive conditions. some of the alleles are not dominant or recessive alleles. And one of those is this condition here, codominance. Because in case of ABO blood typing, either you are A type or you are B type or you are AB type or you are O type. So that means there are more than 2 conditions. And those more than 2 conditions are because the alleles that we inherit are codominant, meaning that if there are 2 different alleles present, those 2 alleles, if 2 alleles are present, then both of those 2 alleles are expressed. One is not dominant over the other one. So for somebody who has A type, so it could be because of this or because of this. Somebody who is B type, so 2 alleles that you have are either these 2 alleles, both of those are B type, or it is because of this. Somebody who's AB type, so it is because you have 2 different types of alleles. And that's why you are not either A or B, because you are both of them at the same time. Somebody who's A type, O type, you actually have none.
Yeah.
So these are the mutants. And that is the majority of the world population. Now O type is the type which is the most common in the world, not only here in the United States, but everywhere else in the world too. These people are actually in good place because these are actually not in good place.
No, because they
These
can
are disadvantaged.
Yeah.
These are universal donors.
They donor, but they can
Because they can donate blood to this group, and to this group. These people are actually in a good place because these are universal recipients. They can receive blood from anybody. Because they can receive blood from here, from here, from here, and from here.
Yeah.
But they cannot donate to anybody. So O type are actually more vulnerable because always whenever there is a need to find blood, it's usually for those people who are O type. Other things we can just ignore. In case of humans, it is difficult to find out what happens because we cannot. In case of plants, we can cross, to the plants to each other. You can actually, even in case of animals, you can do the same thing. So in case of humans, the pedigree analysis is done. Pedigree analysis is based on kind of finding out who your parents were, who your grandparents were, and who were their parents, and all that, and kind of trace it back from wherever the problem started. For example, if there is a problem in me, what they will do is, if I'm here, they will try to find out, the genotype of my parents and the genotypes of their parents, from mother's side and from the father's side, and then see where that disease came from. So, because a lot of these genetic conditions don't start right away. They start somewhere, a few generations ago, and then they are passed on from generation to next generation, and then eventually they become problem. Or at some point, if it is a recessive disease, then recessive disease or recessive alleles will get recombined and will be expressed at some point later on in the generation.
When it skips a generation?
It skips some of the diseases skip generation because, for example, here, so in this case, this person is infected. This infection came, or whatever that disease is, came probably from here and here both. If it is a recessive disease, it came from here and here both of the parent. This one actually had the 2 alleles, and this one had only 1 allele. So that's why half of their children have this infected, whatever that disease is, and others don't have it. Then in some cases what happens is one generation does not show that at all. It does not, it skips one generation because it is a recessive disease, and when the parents are heterozygous, then it skips one generation and it reappears in the next generation after that. In this case, this is the condition in which the 2 alleles are the same, but their children actually, these 2, this one is normal, so it's probably carrier. This one is affected. So 2 of their children have this condition, but others don't have it. So if this one actually was also carrier, this one was also carrier, then you probably will see it skipping some generation and reappearing in the later on. wherever there is a recombination of the recessive allele, it will show. So this pedigree analysis is a kind of a quick way of analyzing and seeing where the diseases come from and what are the chances then that you will be passing it on to the next generation. So in some conditions like hemophilia, for example, or in case of sickle cell or many other diseases like that, it is very important to find out if it runs in your family, then it is very important to find out what type of genotype you are. Because if you are a carrier of hemophilia, or if you are a carrier of sickle cell condition, then and if you marry to somebody who is also a carrier, then the chances are that your children will have 50% chance of developing the disease by recombining these recessive alleles. So same thing here, same thing here. So when you start dating, or it's important to find out, take some blood cell or something.
They used to do that. You used to have to take a blood test before you could get married, but then they stopped it, didn't they?
It was actually not for this type of test. It was done mostly for the blood compatibility. So because so when we say that somebody is A positive or B positive, so this is based on two different types of bloods. One is ABO typing, and other is called rhesus typing. So the plus and minus, plus or negative, comes from the rhesus type. So somebody could be A positive or A negative. So somebody who's A positive, meaning it has this one type of a carbohydrate on the surface of red blood cells, and it also has a different type of a carbohydrate on the surface of the cell as well, which is what makes it rhesus positive. So rhesus positive and negative, or B positive and B negative, both cases here, negative and positive, is a different condition. So if you take blood from B positive and give it to somebody who's B negative, there will be a reaction in that case as well. So that's why people actually used to see the compatibility based on the blood typing as well. It becomes more important, when the baby is born. So if baby is for example, is positive, and mother is negative, so the baby will actually ruin the chances of the next baby to be born, because it will, at the time of the delivery, the mother will get some of the blood from the baby and will develop some antibodies against the next child. That could be actually a dangerous condition, so there are some medications that are used for that purpose now.